Kaitlin Marquis. I'm a graduate student in Rick Bushman's lab at UPenn, and I'm here to talk to you today about my project on HIV replication, transmission, and the metabolome of the female reproductive tract. So I'm really interested in trying to understand how microbial-associated metabolites can modulate HIV replication in the vaginal tract. We know from work done by Duquan and others that women who are colonized by Lactobacillus-deficient, highly diverse microbial communities, which are referred to as HDCs, are at a higher risk of acquiring HIV infection compared to women who are colonized by Lactobacillus-dominant communities, or LDCs. And we know on the LDC side of things that the production of the metabolite lactic acid is really essential to establishing these protective effects of the community. But on the HDC side, we really don't have a good understanding of what metabolites are playing a role in this process. And part of the reason for that is because these communities are so heterogeneous in nature. Much of the existing literature has focused on the production of short-chain fatty acids, but I'm really interested in identifying sort of other metabolites that might play a crucial role in this process. And so with that said, these are the three questions I hope to address in the next 7 minutes. One, are there conserved changes in the metabolome of cervicovaginal lavage fluid collected from women colonized by HDCs versus LDCs, despite the inherent polymicrobial nature of these communities? 2, could any of these metabolites directly affect HIV replication in target cells? And 3, are changes in the metabolome being driven by differences in the microbial metabolic machinery? And so in order to determine whether or not we can find any consistent changes in the metabolome, basically mined literature data to make a heat map where each different row of the heat map here is a different metabolomic study, and each column is a different metabolite that was measured and found to be significantly different in cervicovaginal lavage fluid between HDC and LDC colonized women. And what I want to focus on is sort of this core set of metabolites here that had really consistent changes in at least 3 out of the 4 different studies. And if you look at the makeup of this core set of metabolites, it's actually almost all amino acids, or they're metabolites that are produced from amino acid catabolism, with the exception of the lactate, inosine, and glycerol metabolites. And so then we were interested in trying to, determine whether or not any of these metabolites could actually be directly affecting HIV replication in target cells. So we developed a high-throughput screen based on the TZMBL reporter cell line that produces luciferase in response to increasing amounts of HIV virus replication. And so here's the workflow of the screen. On day 0, we plate the cells, and we added, I believe, over 500 metabolites from our metabolite library. And then we allow that to sit for 24 hours before infecting with the duotropic HIV. And 48 hours later, we conduct a luciferase assay to measure the production of light as a proxy for HIV virus replication. We conducted the screen in duplicate and were able to identify a number of putative enhancers and inhibitors of HIV virus replication. So then what I did was basically mapped the overlap of the results from the TZMBL screen and our metabolomic studies to identify the small number of metabolites here whose concentrations are different between HDC and LDC communities and affect HIV replication in our screen. And one of the metabolites that sort of caught our attention early on is this 2-hydroxyisovalerate molecule, which has been proposed as a biomarker for HDC communities, as it's found in increased concentrations, and it increased HIV replication in our TZMBL screen. So we followed up on these results by treating primary CD4 T cells with this compound. On the left, I'm showing you differences in the amount of extracellular P24 of anti-CD3 stimulated donor CD4 T cells. So you can see that we found a dose-dependent increase in the amount of P24 in the supernatant of 2HV treated cells in a similar manner to our positive control sodium butyrate. And we also saw this when we took unstimulated CD4 T cells from 3 different donors and treated them with 2HV and infected them with the R5 tropic virus. So here we're using flow cytometry to quantify changes in the amount of HIV P24 positive cells. And you can see in orange, again, at higher concentrations of 2HV, we get an increase in infected cells, again, in a similar manner to our positive control sodium butyrate. And so with that said, what we've seen so far is differences in amino acid content between HDC and LDC communities, especially this 2HV molecule. So we were actually really interested in trying to determine whether or not this could be due to differences in microbial metabolic machinery. So we explored the number of amino acid biosynthesis genes between bacterial species that we can actually grow in the lab that have been isolated from either LDC or HDC women and are associated with HIV protection or acquisition. And actually what we found is that vaginal lactobacillus species tend to have much lower numbers of amino acid biosynthesis genes compared to their HDC and HIV associated counterparts. And furthermore, if you look particularly at the branched-chain amino acid metabolism that produces the R2-hydroxyisovalerate metabolite of interest, you could see from our metabolomic study that you actually can see a decrease in the amount of this parental branched-chain amino acids and a corresponding increase in the amount of catabolites produced by branched-chain amino acid metabolism, suggesting this pathway might be important for regulating HIV replication in this setting. And furthermore, it was also sort of interesting to note that there's sort of an overlap with lactic acid metabolism, and that the LDHA enzyme is involved in branched-chain amino acid catabolism. And so what we're working on now is actually developing a flux balance model of metabolic networks from personalized in silico reconstructions of vaginal microbiomes. So we can actually reconstruct individual microbiomes using 16S data, map them to a database that has done metabolic reconstructions, and we can actually model growth and metabolite cross-feeding among different communities and sort of identify metabolites that might be really important for this process. And our plan is to integrate the results we get from our dry lab efforts with our wet lab efforts so we can continue to cross-reference the results from the dry lab and our high-throughput screen, and then follow up by determining HIV replication kinetics and immune cell phenotype of our infected cells. And even we want to get into measuring metabolites of interest in bacteriological culture media and vaginal fluid. So with that, I would like to thank everyone who's been involved in this project and for our funding. And thank you to the organizers for giving me the opportunity to speak to you today. With that, I will take any questions. Thank you.